Introducing a 2025 Cell Stem Cell Review on how bioengineering is reshaping neural organoids. The thesis is simple but powerful. Biology has taken organoids a long way, but the next leap in fidelity, maturity, and function will come from engineering the cell's physical and chemical environment. Neural organoids are 3D tissues grown from human stem cells that recapitulate the developing nervous system. Cell type diversity, organization, and emergent activity. They've driven real discoveries, human-specific brain evolution, mechanisms of microcephaly, and disease models for schizophrenia and Alzheimer's. But the authors are candid about the limits. Today's organoids lack defined anatomical axes, mature slowly, often over 3 months, are poorly reproducible batch to batch, miss key cell types like vasculature and microglia, and can't yet be recorded across the whole structure. These are the gaps engineering aims to close. The first big idea is controlling where signals appear. In a real embryo, the neural tube is patterned by morphogen gradients along 2 axes using WNT, SHH, FGF, and retinoic acid. But in a dish, we dump those molecules in uniformly, so organoids get no real axes. The review surveys fixes. Microfluidic devices generate stable long-range gradients. A WNT gradient alone can pattern forebrain through hindbrain in order. For finer control, researchers use morphogen-soaked beads, engineered signaling cells, light-activated caged molecules, and optogenetics. The authors argue the future lies in combining long-range and local methods. Signals aren't the whole story. Physical forces matter too. Tissue geometry shapes development. Complex folded organoids follow more brain-like trajectories than smooth spheres. The extracellular matrix is a second lever. The workhorse matrigel is undefined and varies batch to batch. Researchers now tune stiffness, apply mechanical stretch to promote floor plate patterning, and use engineered scaffolds, PLGA fibers, porous silk, even 3D printed vascular-like channels to improve oxygen delivery, maturation, and reproducibility. Once you've built a better organoid, you need to listen to it. Standard tools fall short. Calcium imaging is slow and phototoxic. Patch clamp and shank probes sample a tiny region. Flat electrode arrays only touch the bottom. The frontier is soft, 3D mesh bioelectronics, serpentine and kirigami-inspired designs that grow with the organoid and record chronically across its surface. This leads to a provocative application. BrainAware, where an organoid acts as a living reservoir for computing, performing tasks like speech recognition. The motivation is striking. The human brain runs on about 20 watts. A comparable artificial network needs millions. Biological networks could inspire energy-efficient, adaptive computing hardware. Naturally, this raises ethics. The authors address the headline worry directly. Organoid consciousness is not a near-term concern. These tissues lack the size, architecture, sensory inputs, and cell diversity required, and mature only to roughly second trimester stages. The immediate issues are practical. Donor consent, especially if organoids are used for computing, something donors never imagined, and they advocate building design ethics into engineering from the start. Looking forward, expand into drug screening and toxicology, improve maturation and add missing cell types, standardize protocols, and keep neuroscientists and bioengineers working hand in hand. So 3 takeaways. First, bioengineering, not just biology, is the lever for the next generation of organoids. Second, the toolkit spans the full stack: chemical gradients, physical scaffolds, and bioelectronic interfaces. Third, greater capability brings a responsibility to embed ethics into design from day one. As the authors close, quoting Emily Dickinson, "If your nerve deny you, go above your nerve."